David is a research director at INRAE in France, the Department of Environment and Agronomy, AgroParisTech.
Yes, thank you. Good afternoon or good morning, depending on where you are. So my talk is about data agglomeration. I will show why it is important to collect data among farmers and how to analyze them. My talk includes three parts. First, I will present three important types of on-farm data. Then I will show why and how aggregate this data. And finally, I will briefly introduce tools useful for data sharing. First, what are the different types of on-farm data available? So we can distinguish three types of on-farm data, namely data collected in experimental farm, on-farm experiments, and farm survey. So what is an experimental farm? So this is a farm dedicated to experiments. So this is not really a commercial farm. Also, this kind of farm can sell their products. The objective is not necessary to earn a lot of money from that. The main objective is to make experiments for research institute or extension services. And the data collected in experimental farms show several advantages, like high flexibility in the experimental designs and measurements. In particular, you can do long-term experiments in this kind of farm. You can also very well define the controls, have a lot of replicates and so on. You can also have sometimes large experimental fields. And in this type of experiments, the conditions usually are closer to actual farming conditions compared to small plots. However, this type of data suffers from one important limit. Indeed, also an experimental farm is relatively close to an actual commercial farm. It is not exactly the same, and it does not have the same type of constraints, of course. So this is an example of experimental farms located in the Paris area. It's actually one of the experimental centers of INRAE in France. And one of the experiments conducted in these farms aims at comparing 4 cropping systems, namely a productive system, a zero pesticide system, a system aiming at reducing energy consumptions by 50%, and a system aiming at reducing greenhouse gas emissions by 50%. So these 4 systems are conducted in relatively large fields in a factorial design with 3 replicates here. It's a long-term experiment. The data collected for something like 10 years or more, and different types of observation are collected in these systems, like yields, greenhouse gas emission measurements, biodiversity observations, and so on. So the second type of on-farm data corresponds to data collected in real commercial farms. So an on-farm experiment is different from an experimental farm. Here, the data are really collected in actual commercial farms. So an on-farm experiment is typically based on replicated designs, but here usually it's done only on 1 year, sometimes 2, but not much more. And this type of data has several advantages. So it can include the control, which is of course very important. It can include a few replicates, not a lot, but usually we have 3 or 4 replicates. Another big advantage is that most of the interventions are done by the farmer, that is under realistic conditions. And of course, this type of experiment involves strongly the farmers, which is always very good to spread the knowledge and to exchange ideas with farmers. But here as well, there are several disadvantages. First, the experiments are not always fully randomized. Usually you only have 1 year of experiment with a few exceptions. Another issue is that only some specific practices can be tested by the farmers. And yes, as I said, this type of experiment is not very well designed for long-term studies. So this is an example of a network of on-farm experiments here located in Iowa. So each point corresponds to one farm and to one experiment. So each experiment aims here at comparing the effect of fungicide application on soybean yields compared to untreated controls, and each trial follows a replicated strip trial design with 3 replicates, and each replicate includes a control and a treatment, and each experiment is directly conducted by the farmer. This is a second example of a network of on-farm experiments. So here it's in France. So some of the experiments were conducted on soybean, they correspond to the orange points, while others are conducted on sunflower in green. Each experiment here compared 2 plant densities, one high and one low density, and the objective was to study the impact of these different, this contrasted densities on crop production in dry conditions, to see whether in dry conditions it's better to increase or reduce crop densities. And the third type of experiments correspond to farm survey. So here there is no treatment, no control usually. The observations are directly collected in real farms under current farming conditions. And here as well, there are some advantages and disadvantages. Advantages, it's possible to get very large sample sizes. And the data here are collected under actual farming conditions, and you also can involve a lot of farmers here. But the disadvantages is that you only have usually one type of system, of cropping system available in each farm. You have no control and no actual replicate. This is an example of on-farm survey. So here it's a relatively large survey, including more than 900 non-organic arable farms in France. So you also see different type of measurement observations were collected here. I only show here on this slide the treatment frequency, which corresponds to a number of normalized pesticide treatment per season. So on the right, you can see the distribution of the treatment frequency index across the whole group of farm, and on the left, you can on the right, sorry, you can see the farm-specific value. The red indicates high number of treatments, like 8 or more, and the green indicate low values. So, as I said, these types of on-farm data have their own advantages and disadvantages, and there is a trade-off actually between the proximity to the real world and the risk of confounding. So confounding is often referred to a mixing of effects. The effect of the factor under study on a given outcome can be mixed with the effect of additional factor, resulting in a distortion of the true relationship. So here I show a trend. Of course, there are some variability, but overall, we can probably say that experimental farms are closer to real world than lab experiments and small plot experiments, but tend to be a bit less close to real world than on-farm experiment and farm survey. On the other hand, the risk of confounding is often higher on on-farm experiment and on-farm survey because of relatively short time coverage, of lack of randomization, of inappropriate control, or even because of the absence of control in farm surveys. There is actually a second trade-off, but now it's between proximity to the real world and design flexibility. The main advantage of experimental farm is that they offer a very high level of flexibility in terms of experimental design. This flexibility tends to be lower in small plot and to be higher in small plot and in lab experiments. But the flexibility tends to be lower in farm survey and in on-farm experiments because of the constraints of the commercial farms. So now the second part of my presentation is about why and how to aggregate data. So in fact, it's very useful to assess the performance of different farming practices across a large range of conditions. That's where data aggregation is useful, because you can compare the yields resulting from alternative practices. You can evaluate the economic profitability, evaluate environmental impacts, and conduct multi-criteria analysis. And there are three main approaches to do that. The first one is very close to what is done in meta-analysis. It's mean effect size estimation. The second one is regression, and the third one is machine learning. So concerning mean effect size estimation, the principle is to summarize each individual experiment by an effect size, representing, for example, a relative yield change between an experimental intervention and a control. And you do that for all experiments. You repeat the computation, the calculation of the effect size over all individual experiments to get an estimated individual effect size and a confidence interval describing the uncertainty. So if you repeat that across a large range of experiments, you get an ensemble of individual effect sizes plus their confidence intervals. And the next step is to estimate the mean effect size as a weighted mean of the individual effect sizes. And then you have to evaluate the uncertainty in the mean effect size estimates, and you can describe the uncertainties through two probability intervals, the confidence interval and the predictive interval. The first one assesses the uncertainty in the mean effect size estimate, and the second one is able to allow you to calculate the predictive interval for a new situation. So just one example, this is an example of 15 long-term cropping system experiments on maize and wheat in Europe and United States. What we got from this network is a yield ratio. So we have a series of yield ratio and their confidence interval, and from that we were able to compute a mean yield ratio. So here it's a yield ratio that allows us to estimate the yield gain or loss resulting from a reduction of the chemical input in the cropping system. So, so here we compare conventional high input systems with low input systems, and we found here that we have a ratio of close to 0.9. So about 10% of yield loss in average, with some variability across experimental sites. Now I present a second example of on-farm network, including now 19 trials in Iowa. The objective was to assess the effect of higher seeding density on soybean yield. So the seeding density was increased from 18 to 21% compared to common seeding density. And here again, we computed the ratio of yields. And you can see again the distribution of the individual yield ratio across the different on-farm experiments. And from the statistical analysis based on the Bayesian statistical model, we found that there is a 28% chance that a high density on soybean will not increase yield, showing that in fact this treatment has a relatively low level of effectiveness. Another type of approach to aggregate data is regression. So everybody knows regression, of course. It's the principle is straightforward, but the interpretation is sometimes difficult. So of course, the principle is to relate response variables to one or several inputs. For example, it's possible to use on-farm data to relate the effectiveness of disease treatments as a function of the disease pressure measured in the field. This approach was recently used to assess the effectiveness of two biocontrol methods against botrytis in organic grape wine in France. So this study is based on a network of on-farm experiments assessing the effectiveness of these two biocontrol techniques as a function on the x-axis of disease pressure in grape wines. The results were obtained here from a generalized Bayesian statistical linear regression model. Clearly here we see that the disease incidence on the y-axis was much higher in the untreated control in orange here compared to 1 yard treated with biocontrol techniques. The last approach that I would like to introduce is machine learning. So machine learning becomes more and more popular now. The objective is to train non-parametric algorithms from a large data set. And based on the trained algorithms, what we do is to predict the response of interest, for example, yields of or greenhouse gas emissions or something else. And then you do that with different type of algorithms, and you compare the predicted value to a test data set, and then you are able to find the most predictive algorithm. So why machine learning is powerful? Because machine learning is able to find a very good balance between bias and variance, and is able then to minimize prediction errors. So we have many methods available to do that, and the good news is that it's now relatively easy to run these methods with specialized packages. Just a quick example. So here we use a network of experiments in 1 yard in the Bordeaux area in France, where incidence data were collected across several years in untreated plots. And based on that, we were able to train several machine learning algorithms to predict the occurrence of high mildew severity as a function of different inputs related to temperature, rainfall, and disease onsets. And from that, we were able to derive predictive tools computing the probability of high severity as a function of various weather data, in particular the rainfall in May. So now I'm almost done, and I just want to spend 1 minute on different recent techniques proposed to share data to improve data sharing. So how to share data? So of course there are many different ways to share data, but two recent tools appear to be particularly relevant and useful in practice. The first one is the data paper. So data paper is a paper attached to a public open access database. So the purpose of a data paper is not to present new findings, but to describe a data set made fully available to the whole scientific community. So it's very relevant to share data in a transparent manner, because the data paper explains the origin of the data in great details, and as the data set is attached to the data paper, it's very easy to retrieve the original data and to reuse them for other applications. So here it's an example of large-scale on-farm data collected across the world on conventional tillage system and no-tillage systems. It's an example of data paper presenting a big data set, including data comparing the performance of different type of grain legumes on yields. And here again, another example of a big data set collected on farm, including yield data for major lignocellulosic bioenergy crops. And a second type of tools is a tool for data visualizations. So it's a very relevant approach to share data. So these tools, they do not only provide access to large data sets, they also allow users to explore the content of the data sets interactively, user-friendly tools. You can locate the experiments in space and time. You can describe the distribution of the observations and the relationship between the measured variables. So some of them were developed in America, like this one called Fast and maintained in Iowa, and other were developed in Europe, like this one, which allow users to compare the performance of different types of cropping systems. So I'm finished. Thank you for your attention.